Hi, everyone, and thanks to the EBI training team as well for giving me this opportunity to run this webinar along with Coleman on the introduction to the COVID-19 data portal. So this is going to be a little bit of a whistle-stop tour of the data portal and the data platform. I'll try and cover as many things as I can. And if you do have any sort of specific things that you are interested in, then hopefully this will form a really good introduction to those. So in terms of what will be covered today, I'll talk about the data platform, which includes a bit of background, context to it, and briefly talk about the three main components of the platform itself. Then I'll focus in a bit more and talk about the one of those components, which is the COVID-19 data portal, which this webinar is based on, and in particular around the usage of it. So covering things like submissions, searching data, retrieving data, and visualizing data as well, with a couple of examples of full integration of a couple of tools in collaboration with the VO project within the data platform. But just as a quick sort of poll, I guess it would be great to understand and know if those joining today are actually users of the portal. I think Anna is going to add a poll, in there, and I'll give you a second or a few seconds just to answer that. So we'll be silent for a bit. Okay, I can't actually see the results, so I'm going to assume that there have been quite a few.
Yeah, there have been lots of people answering, so I'm going to end that now.
Cool. Thank
you. Hopefully you should be able to see the results.
Yes, I can now. Okay, that's interesting. Yeah, so actually the majority of people are not users of the portal. So yeah, this will be a good introduction. So thanks for doing that, Paul. And yeah, we'll begin now. So getting started with some background here. The European COVID-19 data platform itself was launched in April of 2020. Here you can see that it's been endorsed by the European Commission. So there was a bit of press around the platform itself, and it formed part of the response by EMBL to the COVID-19 pandemic. And what you can see here is the data portal itself, which is kind of the entry point to the platform. And here all sorts of different types of data that are presented in one resource that is then available for scientific researchers downstream. So the data platform itself provides open and rapid access to not only data and data sets in the portal, but also tools and workflows, other resources, and they might be popular within the community, for example. It builds upon open standards. So here, this can be around the actual data types, metadata. It can be around those tools and workflows, and generally within the infrastructure as well. And even though the data platform is European by name, we actually see that data is coming from all over the globe. And not only that, but people who are accessing the portal and platform are also coming from all corners of the globe, which is really, really good to see. And together, what this is kind of hoping to achieve is to enable diverse scientific research to fight COVID-19 and try and sort of plug itself into certain parts of, for example, vaccine developments and drug repurposing trials, or generally to better understand SARS-CoV-2 itself. On the right-hand side here, you can see the various different projects that have been involved and are involved in the different aspects of the platform. So there are many different activities here. as part of the platform. So it is a very highly collaborative effort, and it's really good to see what can be achieved with this collaboration. So briefly, there's the three main components. I'm going to talk mainly about the data portal in a bit, but just to mention the other two, which include firstly, the SARS-CoV-2 data hubs. So these are essentially a workspace and a toolbox that enable users to share their sequence data in a private pre-publication mode before they're ready to release it into the public space. This can also is also particularly useful when groups are part of consortiums and closed groups who wish to share and check data before they're actually ready to release it. These data hubs may also include tools to analyze and visualize their data and sort of bring those to the actual data sets and data hubs. And some of these aspects are already being used as part of the systematic analysis of public raw data within the COVID-19 data portal. And so this is in collaboration with the VO project, and we're producing variant calls and consensus sequences that are calculated and worked out from the analysis. And this is important to mention as some of the downstream visualization services, for example, and is another large data set being available for consumption by scientific researchers. The second component here is the Federated European Genome Phenome Archive, or EGA, and this has been leveraged to support sensitive human data sharing and provides restricted and controlled data access, which you can kind of make out in the middle over here. So this involves requesting access via data access committees to gain access to data and setup of appropriate data sharing agreements. As the name suggests, it is a federated data model. So here is an example. You have a local EGA instance, which is essentially a node in another country. And therefore, the data sets themselves do not leave the national borders. And there is just this metadata synchronization that occurs with the centralized EGA node. This means that anybody who does want to access data held within the local EGA instance, they would need to access a request access over here. So part of the reason why we've been able to act relatively quickly as part of this, the response to the pandemic, is that we had already established foundations in place. And this included at the top right and continues to include the EMBL-EBI resources, or Elixir core data deposition resources at EMBL-EBI. And they span across different themes and present data types within and help to present these data types within the COVID-19 data portal. You can kind of just about make out ENA in the molecular archive section here. Which is the team that I'm part of. Underneath here are the compare data hubs. So these are the precursor or foundations for the SARS-CoV-2 data hubs. There's more about the compare data hubs in this paper that's linked in the slides. And essentially it includes, for the SARS-CoV-2 data hubs as well, similar aspects and components such as the submission services, the analysis components, including tools and workflows or workflow management and infrastructure, and then the search and retrieval services and portals, as well as the visualization services as well, and bringing these aspects to particular data hubs and therefore the data sets. The middle here are the Elixir member states and collaborators and network. And this enabled us to find and understand more about the national sequencing efforts within Elixir member states. And not only that, exchange knowledge and around tools, data sharing, and so on. The bottom here are our collaborators from other pathogen projects. And they have been really important in supporting some of the scientific decisions made as part of some of the activities surrounding the COVID-19 data platform. And then the European Open Science Cloud, which is a wealth of resources or knowledge about resources and services being brought in one place here. So you can see, again, highly collaborative. So that kind of covers a really quick background on the data platform. And now I'm going to sort of zoom in and focus on the data portal itself. And I'll start off by talking about submissions to the data portal. So to submit data to the data portal, there is a dedicated submit data page that you can view at the URL that you'll see there. And there you'll find documentation regarding how to actually submit the type of data that you wish to submit. It currently spans across 11 different data types, and for each individual data type, there may be multiple data submission routes available, which users have the option to pick and choose from. The data submissions occur with the underlying EMBL-EBI resources and databases, which then return and feed back the data to the portal front end, which is what you're seeing in the data portal. So as you can see, there's quite a number of different types of data, and so instructions may vary depending on that. But just to give you an idea, I thought I'll talk about how to share sequence data at the European Nucleotide Archive, or the ENA, as I'm closely affiliated with it. But if you do have queries around other data types and how to share those, apart from what's in the documentation, then feel free to contact us on the help desk, and we will reroute your query to the experts at the particular databases concerning your data type. So in terms of submissions to the ENA and sequence data, we have a set of standard tools, such as the Webin family of tools, including ways to interactively submit your data via the Webin submissions portal, which is the interface. Then there's a programmatic option via Webin REST, which enables users to prepare and share XMLs and submit those to the Webin REST API. And then Webin-CLI, which is a command line interface tool that enables users to pre-validate their data before they actually submit it. And therefore, it enables them to identify any errors prior to the submission stage. Then we have the drag and drop uploader tool. And this, as the name suggests, is an interface that enables users to drag and drop their metadata along with their sequence or data files into a box on the interface. This is currently optimized and more suited to smaller scale submissions. And so we do recommend that users contact us via the register button as on the documentation. And then one of my colleagues will be in touch to guide you through the process. Aside from the actual tools that are used to submit data, there may be other things to consider. And so, for example, here at the ENA, we use a metadata model to help construct and provide structure to the the data that's being submitted, to make it contextualize later on downstream when pulled and retrieved. So this metadata model, you can see, is linked at the text here. So if you are interested in submitting data to the ENA, we do recommend reading up on the metadata model. Aside from that, we do use specific metadata standards, and again, this is in the form of metadata checklists at the ENA. So these present a set of mandatory fields, a set of recommended, and then optional fields, and then if there aren't any, a user-defined field can be included within the submission, but we do recommend contacting us first to use appropriate terms and such. And this helps to contextualize the actual metadata around the biological sample. And you can see a few examples that I've included depending on the source of your data, whether it's pathogenic or from a metagenomic background. Couple of screenshots here that will show you what I'm kind of talking about. So these are mandatory fields within the two checklists, or a couple of checklists out of the three I mentioned. And there's detailed documentation within the SARS-CoV-2 submissions guide that details not only this metadata step, but also how to submit and what may be suitable for you as a user. So yeah, this is all documented in this guide quite extensively. But if you do have any questions, yeah, feel free to contact us. We also have this submissions workshop. So this includes a workshop with videos, instructions, and demo data to test SARS-CoV-2 submissions accessible via the link that you see here. And it provides sort of experience and know-how about how to submit both metadata-related objects, but also actual data files as well to couple with that metadata. And in addition to the standard tools, there may be additional tools that are developed and made available to users, depending on what we see, for example, within the community, or whether there's some user-based needs that are within the community. So these are a few that I've included as examples. Firstly, the ENA bulk WebIncli submission tool. So this is a wrapper tool that sits on top of WebIncli. It enables users to provide a spreadsheet, which then users are able to slightly more bulk submit their data as the tool coordinates the submission on their behalf. The second there is the Web in SARS-CoV-2 Genome Submission API. This is an API where users prepare JSON objects with the metadata and the sequence data in the same JSON object and submit those per sample. And there's really detailed documentation here, for example, even about how to script these. And so it's particularly useful for those who are submitting data in bulk, and in particular SARS-CoV-2 genomes. And the last example there, so we've been aware that people have been sharing data to GISAID, and this has been really useful and helpful within the pandemic. And so we wanted to make it easier for users to also share data with us. And for this, we created this spreadsheet converter tool, which enables users to convert their submission and particularly provides a metadata mapping between submissions at GISAID to ones that are compatible at the ENA, and this is via the sample XML. And then we also enable users to provide a GISAID accession ID, and this is a user-defined field within the sample attributes. So users are able to link between a soft link between data in the ENA and also GISAID by providing this EPI ID as a value. So yeah, that's a quick tour of submissions for the ENA. And again, to reiterate, other databases may have other ways to submit. There may be less or more. So please do check out the documentation and let us know if you have any queries. So I'm going to switch to search and retrieval of data, and I'm hoping to actually be able to do a live demo as well within this. So there are a couple of main ways to search and retrieve your data. And firstly, this is interactively. And so I'm just going to go into my browser. And typically what you would find when you go to the COVID-19 data portal is this format, it's pretty much the same depending on whatever data type that you're looking at. But straight away, what is catching your eye is this search bar at the top here. And here you can provide quite structured queries, such as, here looking at all the sequences of B117 lineage, or sequences tagged as Omicron, or you can actually search specific, just general text within there. And once you hit search, the underlying metadata table will update depending on the query that you are putting in the search bar. I'll provide an example in a second. The other way to search and sort of query your data is via this left-hand side. So you can see here, these are options and fields that you can query on. So these are what we call facets, and you can kind of select depending on what you're looking for. So for example, things like collection date and the country. So let's have a go at doing one right now. So let's look at, or look for sequences coming from South Africa. And as you can see, I've selected that, and now the table has updated to only show depending on that query. You may not be wanting to retrieve or see all the fields that we're seeing here, for example, or you may be interested in additional fields. You can actually change the view of this table by clicking on Edit Table View on the top right. And so, for example, I'll take away the cross-references, the actions, and add the release date. Hit Apply Changes, and then your table updates, and you can see that last column with actions, for example, is gone, and so is the cross-references. So that's how the table updates itself there from your query. Through the search bar, the equivalent query will look like this. So I'll put country, South Africa. And you can see the same number. It was 3,972 sequences. When you click on it, it will take you to the table. And because it's kind of remembered that I selected this table view. So that's an important way to query. If you are interested in creating more structure, even more structured queries, so making it look a little bit more like an SQL query, for example, EBI Search provides this really handy tool called Query Builder that enables you to create a particular term that you can query on. So I'll kind of show you an example. So here, under the domain, you have all the different types of data. There's so many there, but the search bar is very handy here. So let's look at sequences like I was looking at a second ago. Select sequences COVID-19, and then we can change and then put our query in. So let's put first country, South Africa. In addition to that, let's add another rule. And here you can select whether it's an and or a not. We need it to be an and because we want to look for all sequences coming from South Africa that are tagged as Omicron in the WHO nomenclature. So what you see underneath here is the query preview, and you can see the string there. But to copy and paste it, just hit search and then copy that, and then you can paste it here. And you can see the number of results has updated to 884, and that's the table there. So yeah, this is a really handy tool. I would recommend it. It is linked in the slides as well. So once you are happy with the table that you are querying and or data that you're querying and that you're happy with the table, you hit download. Actually, I should also mention that you can select all of this data, or you can select a few, and then you hit download, and you can download either the actual data files or the metadata for the selected results or all the results within the selection there. So in this case, with sequence data, there's a couple of different data formats. There's EMBL flat files or EMBL flat file formats, and then FASTA, which is just the sequences. So you can select that and hit download. Alternatively, for metadata, you have just the list of accessions, so that first column, or the entire TSV of metadata there. And then you hit download and it'll download that for you. So yeah, that's how to do things interactively. I'll just go back to my slides just briefly. But to do things programmatically, there's a couple of tools that you can use. So here I'll talk about the data portal bulk downloader tool in a second. but there's also the API. And for the API, again, I'll go back to my browser. When you go to the link that was in the slide there, you will see this page. So here you've got information about the parameters and then the endpoints underneath, of which there are more than 40. And this correlates to the type of data that's held within the view that you see in the portal. So for example, for sequences, I copy and paste this endpoint here. For query, again, I would recommend if you're not sure about your query term, EBI search is really handy here again. And so you can get your query string and apply that for the query section. So yeah, there's there's a few other links that you can look through. But if you have any questions about searching and retrieving data, then yeah, by all means, again, feel free to contact us. So I'll go back to my slides. So the other aspect that I kind of touched on here is the data portal bulk downloader tool. So this is a really neat tool that is command line based, but very intuitive and sort of mimics the view that you find in the portal in terms of the separation of data types. And what this is really useful for is to enable downloading of a range of different data types in a range of formats, so the actual data files themselves. This is quite extensively documented at the link that you see there. And a few really useful features of the tool include that there's a non-interactive mode, which creates a script for you to then easily download the data or run the query again later on. And you can also reattempt downloads if they do not complete. And by default, the tool automatically retries 3 times and picks up from where it last left off. And the last thing there is that a really cool feature here is that once you run a particular query again or run the tool again, only new and updated files are downloaded. So it helps to keep a really nice local, neat repository for something that you might actually be interested in specifically. So that's searching and retrieving data. Again, a quick one there. I will now switch to talking about visualizations, and much of this work has been supported by the collaborators within the VO project. So both the development work, but also their integration within the EMBL-EBI infrastructure. So this includes, for the phylogeny, integration of Evergreen and PhyloVis workflows. So these are the underlying workflows to create the phylogenetic trees, but also how to view the front-end report that you will see if you go to the link here. The code is available in Bitbucket, and the workflow, which is integrated at EMBL-EBI, is run daily to generate an up-to-date tree, or at least every other day. The current tree itself is based on all the submitted COVID-19 sequence data or consensus sequence data. So that first option that I showed you in the demo a little earlier. But in particular, it only includes sequences that cover more than 98% of the SARS-CoV-2 reference genome. It is separated by WHO continental regions, and this has been purely to support the fact that there's a lot of data, and it clutters the actual phylogenetic tree. And they include Pango lineage assignments as well. And we're kind of expecting updates to this in the coming months. And this is because of the fact that there's so much data that it is not very usable or navigable at the moment. So keep your eyes peeled for this update that we're expecting soon. But just to give you a flavor of what this looks like, this includes 3 main panels or panes. The top left includes a map, which is interactive. So you can scroll in, you can sort of travel around there, and then hover over the pie charts and click on them as well. And this is showing the geographic distribution of the sequences that have been processed to produce the phylogeny. Underneath the map is a metadata table. So this is also, people are able to filter on this by the headers, as you can see here. So users are able to actually query and look for sequences that they may be interested in. And once they have found that, they can select them or select either all of them or some of the sequences, and they will highlight on the phylogenetic tree the nodes themselves or on the map as well. At the bottom, you have the option to change the tree that is being visualized, so you can go to a different continental region. And then you can also change the color scheme or the field that the color scheme is based on. So by default, this is the Pango lineage, but you can change it to things like, for example, country. On the right-hand side here, you can, this is the main phylogenetic tree. You can right-click on either the tree nodes or the background, and there will be additional options that show up where you can either export the tree, you can rotate or view the subtree, or reroute it altogether. So yeah, updates expected there. The next and last thing that I'm going to talk about is the Covio variant browser. So this, I will actually go into the browser itself in a second, but just to mention again, this is includes the integration of a database and front-end application. And this is what you're viewing, this front-end application, what you're viewing at the link that you see there. The app itself ingests variant calls that are produced from that systematic analysis of the raw read data that I mentioned as part of the data hubs a little bit earlier. And it couples this with the variant profiles for variants of concern and variants of interest. And together, what this is doing is it's presenting summaries and graphs that show the global distribution of sequences and lineages. So again, I will switch to my browser. So as you can see here, when you go to the viral sequences page, there is a button called Variant Browser. You can click on that. It may take a minute to load, but once it has loaded, it is very quick and responsive. When you first go to the app itself, you are greeted with information about the number of samples that are derived from a particular country. So you can see the plots are very interactive. You can hover over them. You can deselect specific countries there in this plot. So I just remove United Kingdom, and you can see the spread of, or the number of samples coming from particular countries. And this is a more European view, but you can also view a world view here by selecting and adding countries from this drop-down list here. And again, there's all sorts of other options that you can kind of click around and view as well. The second thing about samples is the maps. So here it is showing the geographic distribution of where samples are coming from. This is the European view. Again, you can hover over, but you also have the world view as well. And again, you can hover over these. Then you have three sections on variants. So the first one here is a country by country view of the weekly cases of particular variants that may be circulating within a particular country I mentioned. So I'll deselect that, and you could see, for example, what is circulating at a particular time point. And again, you can select different countries there from the drop down. Then you have the actual variant of interest view. So here, for example, looking at all the beta sequences or variants. And again, you can kind of hover over these. You can remove certain countries from these and look at where there may be the number of samples from a given variant that is detected in a particular country. So here it's kind of taking in the genomic data as well. And then finally, you can look from a country view of the percentage of samples that are derived from a given variant that's been detected in a particular country. So this is the United Kingdom. I'll take off the not analyzed yet here, but you can see then you get an idea of what's circulating at a particular time point from the actual samples as well. So yeah, we do recommend that you feel free to sort of play around with it. Let us know if you have feedback as well. We're always open to feedback, and we can kind of forward that to developers, but also generally about the portal, we're also welcoming feedback from users as well. And you can contact us again via the help desk for that. So that kind of brings me to the end of the webinar. So I just want to say before I close, a huge thanks to everybody involved, crossing not only teams within EMBL-EBI, but also across other institutes as well, and the underlying projects that have been supporting those different aspects of the portal and platform. And with that, I say thank you for listening as well. And this is, again, the help desk emails. So you have the COVID-19 one for virus data flow, general pathogens, ENA path collabs, which is for the ENA related or sequence data. And then you can also follow us on Twitter for updates and information. The second one there, ENA CovStats, particularly presents information on sequence data holdings within the COVID-19 data portal. And it is a bot that automatically does so on a daily and weekly basis. So yeah, thanks again for listening, and I'll take any questions now.
Great, thank you very much. It's a very nice talk and some interesting visualizations at the end there. And also thank you to Coleman, who's been answering some questions while you were speaking. But we have a couple of questions that we'd like to ask now. But if you do have any further questions, please write those into the Q&A box and we can address those. So up first, we have what kind of clinical and epidemiological data is currently indexed within the COVID-19 data portal?
Yeah, so this is definitely with reference to the, if I go back to the portal, the cohorts tab. So this is a sort of work with the Recoded project, where we are piloting a study to kind of share ClinEpi data, which will involve sharing data at the European Genome Phenome Archive, and also try and also link this with other data types that are more open and shareable across other data types or databases as well. So this is currently in the browser, under the cohort browser itself. It includes a study from the Netherlands, and this is with collaborators in the Recoded project. But yeah, it is we're hoping to also kind of reach out a bit more with regards to ClinEpi data and cohort level data and support data sharing around there. But yeah, we're aware of sort of challenges in data sharing and so on. So we're able to kind of look into supporting that.
Great, thank you. And we just have one more question at the moment. We'll see if any more come in, but is it possible to download the information on the VOCs along time for a particular country?
Good question, and I'm not actually sure about that one. I think to download, you can definitely view it, but I don't think you can download it. That would be quite an interesting feature and might take quite a bit of development for that. But yeah, at the moment, there definitely isn't a way to download it, and we could potentially talk to our collaborators about having something that is useful for that.
Worth noting as well that we make the underlying data powering those visualizations available. So I'll pop the link to the project that data is stored under as well, so that you can power your own similar app in a similar way.